final speaker of the day, Dr. Alan Taylor. I had the privilege of meeting Alan Taylor about 2005. And that was when his firm Intech was taking public a company called Hardware. Hardware was one of the LVAD developers. The company was capped at a mere 30 million. I think Medtronic knocked them off at, what was the price, Alan? $1.5 billion, from 20 million to 1.5. Now, Alan, you're not going to take responsibility for all that, but Alan's firm Intech took public a lot of companies with that sort of potential, where they were not suitable, say, to fund in in American markets or elsewhere, and they ended up being quite powerful here. Just before he and I got acquainted, he'd finished his PhD, and I asked him this morning, where'd you do it? It was St. Vincent's in a laboratory that was focused on diabetes research. Taught him a lot about biochemistry and so forth. So Alan was there for a long time, moved on about the same time as I was beginning my exit out of broking. Puts out the word, who wants 8 years worth of talent that I've built up. Clarity Pharmaceuticals picked up put their hands up. It's fair to say that Alan single-handedly saved Clarity from oblivion, and ended up with a pretty powerful IPO last year. How much did you raise, Alan, in the end? $92 million. When I was in broking, that was the sort of IPO you just couldn't do, and it shows you a lot about how this sector has developed. And Alan, in common with all the other speakers today, have been part of the set of true believers that have built up that capability so it can do that sort of work. And as he'll share with you, he's one of a select group of companies alongside CU6 we heard from this morning that are working on the whole radiopharmaceuticals area. Here to share with us how he's going to change the world in radiopharmaceuticals. Please welcome Dr. Alan Taylor.
I'm just joking. Hi, everybody. Firstly, thanks for your resilience for those here and also at home. And certainly my plan is to make it worthwhile and discussing, obviously, the story of Clarity. It was great to follow some great Australian science earlier. And I want to just touch on James's company. We're right in the same space of nuclear medicine, which is a phenomenal area to be in. And I just want to add to James's presentation that it is great Australian science that has been commercialised. And it's because Australia has this unique advantage in nuclear medicine with ANSTO here, as well as a number of clinicians that are outstanding KOLs globally. And that really allows us at Clarity to progress. Stu touched on other points. He's right. My background is actually originally in science, but moved to investment banking. I do apologise for that. I am better now. And really, the reason for me leaving was because we did all these US deals. Hardware was a US deal, and I wanted to focus on Australian science. I'm not typically very patriotic, but when it comes to Australian science, I think we can do a lot better because a lot of science is wasted just at the benchtop. And Clarity is an example. When I joined, where the science was at the benchtop, originally out of ANU many decades ago, the science started, and it moved to University of Melbourne, where they developed this technology, and now we're in the clinic, and we're at the other end of where Alison is, where she's looking at life when it starts. We're trying to stop it from ending, and that is in cancer itself, and this is in children and adults with cancer. So I'm going to show you some images that you would never seen before when it comes to cancer, but I warn you, there are, when we show you images, there are some of clinical trials that we're running in the US now, giving you the warning of that, but certainly I'm going to show you some exciting data where we're looking to make a difference in this patient population, as well as all the way through and in males and females. So starting off, really our focus is around the commercialization of a new area. So James's product is a diagnostic. We're in the area of what we term theranostic, and that's just a trendy word to say diagnostic and therapy in the one product. So, which is incredibly unique. And we go the next step, that it's actually the same product chemically, because what we really, our secret sauce is our ability to hold copper. You've probably all heard of copper before, and where copper is different is the isotopes of copper. Firstly, copper-64 is a fantastic imaging agent using positron emissions. And then we can also it's called the perfect pairing, utilize copper-67 for therapy, which is a great radiation-based treatment for oncology. So if we can get the product to the tumor, then we have a high-energy isotope to treat. Now, that's the deepest science I'm going to do at this, because I'm going to bring it out now and discuss really the product areas that we're focused on. We're looking at neuroblastoma in kids. I touched on that before. Neuroblastoma is still the biggest killer of kids under 5. It's a devastating disease that we can do better, and we're looking at kids with no other options. We're also moving that into prostate cancer. Prostate cancer, I think I have to tell anybody here how devastating prostate cancer is and how ubiquitous it is. It's such a big market, and we can treat better. And we have 2 products in that space. But one of those products is a pan-cancer target, which also moves into breast and ovarian and those sorts of things. And we're pushing ahead with clinical trials in all of that front. We discussed things, I'm just going to touch on things like patents because people discussed them before and their patent positions. We're at 17 patent families now. So we've got extensive patent position over all of our products and obviously processes, and we continue to build out a patent wall. So we are the only player globally developing theranostics in copper. And really it comes serendipitously from this science that occurred in Australia many years ago. So just building on and I'm just going to touch on the capital markets. What Stu mentioned before is correct. We did the largest IPO ever in the ASX history last year because of where we are as a company and what we're able to do. We raised $92 million. We have just short of $100 million in the bank. When we talk well-funded, that is a well-funded biotechnology company and looking to run a number of clinical trials. The fallout in the markets at the back end of last year towards new IPOs and the like, we're a part of that as once again a broad sell-off for the IPOs, and we find ourselves in this really unique position where we have an enterprise value as low as 70 million Aussie at this point in time for a company of our standing as a global radiopharmaceutical company. And where that is relevant is this area has really come alive in recent years with the development of these types of technologies. And there's been some very large acquisitions when we're talking up to the $2 to $4 billion transactions in companies that aren't not necessarily much further progressed than where we are with single products. And I want to draw out one there as Endocyte, which was a PSMA prostate cancer product that they acquired on the back of phase 2 clinical trial data for $2 billion. And I also want to touch on here Algeta. And the relevance of Algeta is that we're somewhat formed as an Algeta 2.0, that half of our board is now formally Algeta. And that includes our CEO, Colin Biggin, with that breadth of experience in commercializing a product. Their product was Xofigo in prostate cancer. Having all of that capability now within-house is certainly a big advantage when we're looking at prostate cancer and rolling out those clinical trials there. And you'll see where that benefit is playing out when I show you I'll show you what we're doing. Viralytics was mentioned earlier as a successful takeout some time ago. We have a number of people from Viralytics as well, including our CFO and our regulatory component. Good to have them here in Australia, and that was a very successful Australian takeout. So I'm going to try and keep this, once, as I said before, in simple terms. You'll see the product on the left and the product on the right look very similar. And that's because our product, when we develop a product that can target a particular tumor, the product can be used for diagnosis on the left, and then we can change the isotope and it can be used for therapy. So as I said before, you would have never seen anything like this before because you don't typically image therapies and the like, but you'll see on the left here, this is our diagnostic product identifying tumors in a child. So, and then when we find out where those tumors are, we select that patient. So that means we're highly accurate when we provide a therapy. And then because we're using the same product, when we provide the therapeutic product, we hit exactly the same tumor, and we can actually image our therapeutic product, and we can see that we've hit exactly the same spots as the diagnostic. So that means it's both highly accurate and highly precise in a patient population that has no other option, and we know those tumors are radiosensitive. So we're progressing that trial now, as I mentioned before, in 5 centers in the US, and rolling that out very quickly. We're in cohort 2 that we moved to just recently, and it's quite exciting. And these are the other programs, but I want to show you some of the data because that's really the excitement. Seeing is believing was always a cry for us because we can image and then see where our products are going so we can provide therapeutic outcomes. This is our rather extensive clinical trial program and relatively advanced for Australian biotechnology companies. The benefit of my investment banking background was that we could nurture Clarity and do a lot of clinical data as a private company first before listing, hence the later stage that we are of listing as a company. You'll see at the top here where we discuss PSMA, those are diagnostic and therapeutic programs, and we're looking actually to move into phase 3 trials diagnostically as early as next year with that product to get it to the market. I'm going to show you imaging of that shortly. And also in the beginning of our therapy trials. We also have this product BBN, which is Bombacin. This hits another target, and this is a target which is actually ubiquitously expressed. So it's expressed on a number of tumors. So it's called a pan-cancer target, but it provides a great opportunity for us to move into prostate cancer there in actually PSMA negative prostate cancer. We're doing that. We've been doing that trial at St Vincent's Hospital just down the road, which is world-class once again when it comes to nuclear medicine trials with Professor Louise Emmett there, who was outstanding. And then we've got this neuroblastoma work going on in the kids trial. But let's get straight into the clinical uptake, which is really the exciting part of what we do. This is a snapshot of prostate cancer. This is just the US. This is a massive market opportunity, as we told you, let alone, Australia is a small market, but here, relative proportions, prostate cancer is a significant disease. And then when we look at Asia and Europe, we're talking about a massive issue. What we did was something rather novel, and there are some other agents. I mentioned the other agent before, PSMA 617, which was the endocyte transaction. And just to delve a little bit into science again, what they do is use a single targeting moiety. So the target, there's a thing attached to the radiation that finds the cancer. And they use a single one. And what we find in the single ones is that they don't get as much uptake into the tumor and they wash out very quickly. What we did with the University of Melbourne with Professor Paul Donnelly is engineer a new product. So this image up here with the two targets, they're the targeting moieties itself. And what we were fortunate to see, and as Professor Rod Hicks, formerly of the Peter Mac, who helped us out along the way, we got lucky with a product like this where we saw a lot more uptake into the tumor. But we saw two things, not only the uptake, but it stayed there over time. And I'm going to show you a clinical trial where we've actually proven this out in humans as well. And that's why we're so excited about this product. This is this image, and we'll focus on the image here. This is our clinical trial in prostate cancer, in metastatic prostate cancer. This is when the cancer has actually left the prostate and gone throughout the body. This is when it becomes deadly. And so what we can see here in these images is, in the beautiful colorful image here, is that's the cancer all the way down the vertebra there, and we can see throughout the body where that is. And we've actually got this image in comparison to current standard of care in the US, which is a bone scan, and you can see the difference in quality in that. But importantly, in this trial, what we wanted to do was measure over time what our product is doing. If it stays in the tumor, that means we've got more radiation in the tumor to better treat the cancer, and we found that to be the case. So you can see here at 1 hour images on the left here, you can see there's good uptake, exactly the same places where we saw before, but we see it stays in the tumor over time. And that's incredible because what we look at in radiation, the more time the radiation is in the tumor, the better response we have. Okay, so we've passed the first part of this therapeutic trial in understanding what our product does over time. Safety Review Committee has given the go-ahead, and now we're into that next phase of what is dose escalation, because we want to get to the maximum dose that we can provide without having any significant side effects. Remember, the side effects of this disease, though, is death. So what we're trying to do is dose as much as we can, limited side effects or no side effects, so people have quality of life and we get rid of their tumours. That's the plan. These are the studies we're running diagnostically at this point in time. PROPELLER is running in Australia. It's actually pre-prostatectomy. It's early stage, and we're visualizing the tumours in the prostate itself at very early stages. Once again, same to James, using incredible imaging capabilities, we're able to identify where the tumours, for us in cancer, where the tumours are using nuclear medicine. And then we've got COBRA. We've filed an IND to separate it out of our therapy trial in the US, and we've just received the IND from the FDA. It was touched on earlier, the importance of the relationship with FDA. We're in constant correspondence with them, and we got that IND through actually with no questions coming back, which was a unique experience. And so the COBRA is in metastatic disease, so where the disease is moved from the prostate and spread out. So let me show you this. This is actually, once again, this is the PROPELLER trial. This is where it's in the gland itself. We're comparing actually not to what would be viewed as the older technologies of identifying tumours. This is latest technology with another PSMA product using gallium, which has a 1 hour half-life. And we've gone head-to-head with the products just to look at the intensity of what we're able to see with our product. And you can see here it's clear. Images 1,000 words, as they say, and our pictures are, and we can see here our product, lots of product in the tumours, and easily to identify. This gives us benefits then, so we don't necessarily, if the tumour isn't developed enough at the moment, we're missing it, it gives us a greater opportunity of seeing that before the disease grows, which gives us a fantastic advantage. But you can see that, once again, the amount of product in the tumour itself, and that will give us a benefit when we move into therapy. As I told you, the therapy trial in the US, more product in the tumour, the better the outcome. This is the next product that I touched on before, Bombesin, the pan-cancer targets. This really started to take off as a product when we started chatting to Louise Emmett at St. Vincent's Hospital, and really the first opportunity was in breast cancer. And you can see here is a patient with breast cancer. It doesn't look like, obviously, breast cancer here because it's metastasized. It's up in this period here, and we can see it stays there importantly over time and is excreted out of other tissues. This is the important part of a radiopharmaceutical, and obviously was the first human clarification that this was a good opportunity. But what we found when we're running a breast cancer trial is the clinicians at St. Vincent's asked if they could use our product in prostate cancer patients who were PSMA negative. This is about 10 to 15% of this population. And what you see here in the top image is the scan, once again, using this next technology of the gallium-based PSMA product, which is the 1 hour half-life, compared to Bombesin. There was no identification of the tumour with PSMA, but we can see with Bombesin there is clearly a tumour there, and we've constantly had requests now out of St. Vincent's to do a broad range of different prostate cancer patients who are PSA rising but are still PSMA negative. And this is where we'll look to run our first trial in the US. We're looking to file our next IND in the US in the next couple of months to progress that trial there. This is some of the data here, actually from the breast cancer trial itself, though, and I want to show you here. This is the current standard of care in Australia for screening. Here is the FDG scan. You've probably heard of that. It's labeled glucose, and you can see the difference between this this imaging agent and ours. So we will look to expand that patient population as we start to progress those trials in the US to move more broadly out of prostate cancer and then into other areas such as breast, ovarian, and potentially other more deadly diseases like glioblastoma. So you notice there's a little bit of a prostate cancer focus for us, and it's for two reasons. Our products seem to show certainly great efficacy in a prostate cancer patients, not only those who are PSMA positive, but obviously those who are PSMA negative. That covers off a lot of that patient population. But I mentioned in the beginning that we have an Algeta background, and we have on our board 3 people from there, including the Chief Medical Officer from there, the CEO from there, and Colin, who was operations in rolling out in the prostate cancer. And that means we've got a great network of key opinion leaders in the US that allow us to run these trials at all the main centres where you'd want to run prostate cancer trials, and to quickly progress those, obviously, through clinical trials and then to market. But this area here is a little bit closer to my heart, and I'm sure yours. And this is obviously the area I touched on earlier in neuroblastoma. We've used the same images here because I'm actually comparing this to current standard of care, MIBG, which is the imaging agent that they currently use for neuroblastoma. You can see here, this is the same patient visualized weeks apart. You can see the difference between MIBG here and where we can be more specific about where the tumors are and how much to dose, okay? Clinicians are currently trying to make critical clinical decisions based on this MIBG scan. And we're looking to obviously develop that as a standalone diagnostic, and then, as I mentioned, as a therapeutic. Now, you'll see here the regulatory milestones. We've received a few of those to date, including orphan drug designation. But because this is a high unmet need area, kids dying of cancer, they actually have a program called the Rare Pediatric Disease Designation. And this means that if you get a product to market, once you've got this designation, if you get this product to market, you get a voucher to fast track any other drug in your portfolio, and you can trade those vouchers. And one traded a couple of weeks ago for $110 million US. We have two of them. We have one for the diagnostic that will be going against this product here, MIBG, and we have one for the therapy. If we're successful in bringing these products to market, we will sell those vouchers because you get breakthrough status for oncology, which is good, and we'll sell those and whatever the market is at the time. But $110 million US is a lot of money for any Australian biotechnology company. So these are the clinical trials, and I touched on before. I won't go into detail here. We've obviously got the neuroblastoma trial, but this product also has efficacy in other diseases, and this is in neuroendocrine tumours, and we're running that trial here in Australia. When we commercialise the diagnostic and we receive the voucher, we'll look to progress that in new indications which are larger to develop a larger, obviously, revenue stream from that product. Now, I want to talk about because this is obviously incredibly exciting, but I want to talk about how we differentiate ourselves. And the issue with radiopharmaceuticals is that they have what's termed a half-life, meaning that they run out of radiation fairly quickly. And what we do at Clarity is a little bit different in that we have a little bit of a strategic benefit in that the half-life of our diagnostic is long enough so we can centrally manufacture and broadly distribute to wherever there's a PET camera. That's the differentiator for us. And then on the therapy side, we don't rely on nuclear reactors. We rely on linear accelerators that can be rolled out. And there's an aging nuclear reactor network at this point in time, and there's a shift in the US to non-reactor-based radiopharmaceuticals, and we work very closely with a group called North Star, who's developing not only technetium, which is a non-reactor based for James's company, but copper-67. I'm going to touch on these quickly. Gallium and fluorinated products have very short half-lives. We're talking 1 hour to 2 hours. You don't need to be a clinician to know, or a scientist to know. That's probably going to be a little bit of a nightmare when you're trying to make your product and those sorts of things afterwards. Then you've got to treat the patient. Then the patient has to be there an hour later and they have to scan. For copper-64, we centrally manufacture at large volume on a cyclotron. We make the product there. We can broadly distribute, as I mentioned, to wherever there is a PET camera. We're currently doing that in the US and Australia for our clinical trials. And we have a separate diagnostic program to progress that through. On the therapy side, nuclear reactors are required for the production of lutetium at this point in time. And there's two issues with that. One is the aging infrastructure. Most of it's in Europe. Not a lot of it's in the US at this point in time. And there's no real growth expectation in this sort of nuclear area in the US. And there's a shift towards non-nuclear based, as I mentioned. But importantly, particularly with our current global issues, ytterbium is sourced from Russia. And so there's a potential for supply issues on lutetium there. We've seen nuclear reactors go down just recently. There's one down currently in Europe, and there's supply disruptions around that. We avoid all of that. This is this is a Rototron here. You can see how small it is. We can centrally manufacture all of our product. This is in the US now, and centrally manufactured just with one of these Rototrons commercial supply. North Star have significantly invested in a number of Rototrons to build this out for the production of moly. And copper 67 is such a clean isotope to make. There's no long-lived byproducts or anything like that to produce. So we can roll out, this area is barely scratching the surface of a massive oncology market. We can roll this out not only with efficacious products, but also ones that are cognizant of the environmental issues of isotopes. So this is really the focus and summary of what I just mentioned about the shift away from where we were to where we are, which is this new age of theranostics, but really with a far less environmental impact. I'm going to touch on the team just quickly as it's relevant. As I mentioned, there's 3 of these board who are actually formerly of Algeta, which was the $2.9 billion US takeout for a prostate cancer drug. That's Thomas Rundall there, who was the CEO. The starting CEO was there all the way through to the end of takeout, and then he was actually the managing director of Bayer Norway. Algeta was a Norwegian company. We got Gillies O'Brien here, who was the chief medical officer for the phase 3 clinical trial, running multi-centers. That was successful and commercialized, and Colin Biggin, who was part of the operations in that rollout. So we haven't just got people who know what they're doing. We've got people who have done it before and have all of those networks and capabilities. And we have some other people now in the US coming from groups like Cardinal and Algeta to help us expand. We've got Rob Thomas, who was the chair of Heartware, who I've known for a lot of years. He's the former Citigroup head of investment banking there. And we've got Chris Roberts, some of you might know, of Cochlear fame. He was with Cochlear as CEO for 12 years. And we've got Roseanne Robinson here on here as well from ANSTO, our local nuclear reactor. I'm going to just touch quickly on the scientific on board on one person here in particular, actually 2, Paul Donnelly, our inventor of our technology. I'm fond of the science and Australian science, but Oliver Sartor here has been the principal investigator on 4 prostate cancer products, phase 3 clinical trials, and getting them to market. He's known as the prostate guy, and has been instrumental, obviously, in us rolling out in all the main prostate centers in the US. When we're talking about places like the Mayo and the like, really big names in that space. This is the summary. We released this to the ASX this morning because I'm giving a few of these over the next few days. And so let you read it off that as a summary of everything I've said and our contacts. Thanks, everyone.
I actually had a question, Alan. I mean, many people, whether we like it or not, are going to be impacted by various types of cancer, especially in Australia. And just regarding the treatment of tumours in particular, do you see your research and treatments developing into really low survival rate cancers like brain cancer, for example, and treating those type of tumours in the future? Are those sort of things on the radar? Looking into those as well, or?
Yes, definitely. Yes. So, and I didn't sort of use the word before, but we have a platform technology. It's just that platforms sometimes lack focus. So we need to get products to market as a focus, but we can actually attach any targeting moiety to our patented position. So it actually opens up myriad opportunities. And I didn't have time because it was a short amount of time, but we have a discovery program looking at things like that right now. We're looking to bring in new products. We work with a number of the universities in Australia and the US and some in Europe at the moment, looking at what's next. And so glioblastoma is one of those. Bombesin might be successful in glioblastoma. It's a difficult tumor to treat, but we will look to run probably a diagnostic study as part of a basket study for bombesin because it's incredibly important that we do that. And we may do that because we can do that quickly in Australia. Look, if we're hitting the target, that means we can hit the target with the therapy. So that means if it's radiation sensitive, we hope to have a positive, efficacious effect. And that's what we can do with ours because you can see if the product hits or not, select that patient and provide therapy. So the answer, that's a long way of saying, yes, we are.
I'll open the floor to any other questions as well.
in immunotherapy. Can you be synergistic with checkpoints and other stuff that's coming?
We can, yes. So let's just take a backward step. Cancer has been backward in its therapies for many years. You know, it's you're looking at chemo, surgery, and external beam radiation, right? They've all got a bit better, right? But the breakthrough has happened in the last 10 to 15 years, and it's happened in two areas. One is immuno-oncology. So what it does, the cancer grows invisibly in the body from the immune system. You can hit a protein which makes it invisible and stop it from working. So then the cancer is visible and the immune system attacks the cancer and kills it. It's really awesome. The next area is our area, which is targeted therapies, where we can more specifically target the cancer because we know about the biology, okay? Now, the change in cancer outcomes has gone up exponentially because of those developments, okay? With us, immuno-oncology is interesting. It's only having around about a 20 to 30% efficacy rate in things like skin cancer and the like. And so what we have found is that radiation is able to sensitize tumors to immuno-oncology. So that means pairing with an immuno-oncology group or drug or the like, and this is where strategic alignment comes in. And but using those in together, where you would provide a drug like ours, hit the target, provide radiation, make it more sensitive to this other product to increase that efficacy, maybe we can get to 50%. If we get to 50%, that's you got to think that 30 to 50. You think, oh well, it's still 1 in 2. That saves a whole lot of more people's lives, and that's really our goal.
Thank you, Dr. Untitled.